these graduate students are about to present to you represent years of hard work. But there's no harm in having a little fun while we celebrate this great effort. So when each of the contestants comes on the stage, please give them a warm welcome. And now I'd like to invite our first contestant to the stage, Leah Foltz from UC Santa Barbara.
This is my advisor, Dr. Dennis Clegg. And recently, we've had an opportunity to work with this young woman, Lily. Sadly, Lily is going blind. She has a typo in her DNA that's causing the cells in her eyes to degenerate. And as a cell biology lab that studies blinding diseases, we're interested in trying to help Lily. We want to know if we can find where that typo is, and if we could fix it, could we treat her disease? Could we possibly take her disease cells, edit her genetic mutation, and then use her healthy cells as a therapy? In other words, can we personalize medicine to the level of using someone's own cells to cure their blindness? Before I can answer that, we need to go over stem cells. About 10 years ago, scientists discovered that you could take a skin sample from a person and turn those skin cells back into fully functioning stem cells that can become any cell type in the body. And more recently, scientists have figured out how to get into those cells and actually edit a typo. The cells on the right are ones that I made in lab that were originally a skin sample from a patient like Lily, for which we made stem cells, corrected their typo, and are now studying the eye cells in lab. And not only can we make one cell type at a time, we can actually make entire small tissues. This image is showing the cross-section of what is essentially a miniature eye grown from healthy stem cells. And in red, I'm showing where the light-sensing cells are, whereas in green, the protein directly related to Lily's disease. We were actually successful in finding where her typo is, and we now know how to study that in healthy cells. So the answer to my original question is yes. We do have the ability to take a person's disease cells, edit their typo, and possibly use those healthy cells as a therapy. But before we can move forward, we need to make sure that we're making the correct cell type, that we edit the typo efficiently, and that we can safely transplant these cells back into the patient. If we can figure this out for Lily's disease, it will give hope to so many people who suffer from genetic diseases. Thank you.
All right, well, while the judges are doing their judging, let's have a little conversation. Have you always been interested in doing research?
Ever since around my second and third year in undergraduate, I started doing some work on campus at UCSB and just really wanted to stick with it. I liked solving problems through research and went on to join a lab there at UCSB.
So, so let's say when you were in the 4th grade, what did you think you were going to be when you grew up?
I wanted to be the President of the United States because
you'd be good.
I have the same birthday as Abe Lincoln, so I thought that was a sign.
And so when you're not doing research, what kinds of things do you like to do?
Luckily in Santa Barbara, we have beach volleyball right outside our lab, which is great. So that's a really good way to spend time in between.
I'm just imagining all these researchers going out and playing beach volleyball.
Yeah, there's a lot of active students on UCSB's campus.
Yeah. So how do you want to continue your research? How do you think about your future?
I really want to see this type of cell therapy move forward. I know there's a lot of hurdles with stem cell therapy, and I know it's going to be a long road, but I think there's a huge opportunity to make a big impact if we can automate more of this, and we can really get this to the level where any patient could just come in with a disease, have their stem cells made, have whatever they need corrected, and walk out with a transplant would be my pipe dream for this.
That would be,
Yeah.
that would be awesome.
Yeah, we'll get there.
So, is there something about you that, say, your graduate advisor doesn't know?
Oh, Dr. Clegg. He doesn't know that every time we do lab cleanup once a year, we like to use all of our PPE to come up with a choreographed lab dance, which hopefully doesn't get us in trouble with EH&S, and post that to the lab website. But I don't know if he knows that.
Yeah. So can you show us the lab dance?
It was the whip nene was one year. I don't know. I'll leave that for the real dancers. Yeah.
And you talked about beach volleyball, but what other kinds of things do you like to do? Or maybe alternatively, I should ask, how many hours a week do you spend in the lab?
I don't like to calculate that. Yeah, we spend a lot of time in lab, and luckily the lab that I work in is a great group of people, and we get along, and we do hang out outside of lab, which I think is awesome, and we'll go to the beach and go to the local cool breweries that Santa Barbara has, but mostly in lab, yeah.
So, like when you all go out for a beer or what have you, like what do a bunch of lab researchers talk about?
We sometimes have to set a rule not to talk about work because I'll get so excited, talk to one of my postdocs, like, okay, so I think this is the result I got, and then my lab manager will like, no, stop, drink your beers, stop talking. Yeah.
And how has Santa Barbara been in terms of campus and a community?
Yeah, I love Santa Barbara. I went there for undergraduate, and I'm in my 4th year, so I've been there for 8 years. I can't say anything bad about it. I love the community there. I love my advisor, Dr. Clegg. I wish he could be here, but he's busy at conferences, traveling, getting grant money. And yeah, I really like the community there and the support from the graduate division that's here today.
Yeah. So do you have any, you kind of alluded to it, but do you have any concerns about conducting the kind of research that you do in terms of, funding or availability, that sort of thing?
Yeah, I mean, of course, with stem cells in the history of this state, there's been ethical issues that have been covered by federal funding. The California Institute for Regenerative Medicine was a huge grant that allowed our lab to get the research done that I've presented. And I think that keeping that up and having access to that money is huge for being able to have any hope in curing disease for people.
Yeah, and you mentioned in your presentation that techniques had been developed to help you more easily edit the DNA, I guess. Can you talk about that a little bit?
Yeah, so buzzword, we use CRISPR-Cas9, actually, which I know anyone from Berkeley has probably heard a lot about, or anyone who listens to NPR. There's been a lot in the news about essentially using something that bacteria developed over time to cut out the wrong letters and then be able to place the right one in. And we can use that technology that bacteria made over time in cells to edit them.
So it makes it a lot easier to edit the cells.
Yeah, exactly. It's something that was naturally evolved in another organism, so it's sort of perfected, and we've started to adapt it to use in human cells, and using it when they're stem cells allows you to really correct it and grow a bunch of the correct cells that you need.
Yeah.
Which is not something that was always possible. There are other techniques, but it's probably the best one we have now.
Right. So you've talked about the eye. What are some of the other applications of your research?
So the eye is a great place to start with stem cell research because you can see directly into where you put the transplant. You can monitor that, which is really important to make sure that you're not seeing any negative effects. And there's also been a lot of research into paralysis and stem cell transplants into the spinal cord. But if you look through the literature, there's really no disease that doesn't lend itself to stem cell research because whatever part of your body it is that's not working anymore, whether it's your pancreas because of diabetes, or your nerves, or nerves in your brain for Parkinson's, we could potentially replace those diseased cells with healthy ones. So stem cell therapy doesn't end at just blindness and paralysis. It really can apply to any disease.
Yeah. And do you see yourself remaining in academia moving forward, or moving out into the private sector? Or
I'd like to leap into industry. If anyone, no. I'm a fourth year. No. Yeah, I do enjoy academia, but I see myself in industry.
Yeah. Do you see yourself staying in Santa Barbara, or are you going to be portable?
I'm portable. I love Santa Barbara, but I understand that it's small, and there's some great startups there that have come out of some of the management programs there. But yeah, I think San Francisco and San Diego are kind of the polar ends of biotech, it seems.
Yeah, so for anybody watching,
Just kidding.
How do you explain to your family what it is that you do?
My little brother's actually here. So I think that's what was cool about this program, is this was the first time that a lot of my family actually understood what I do all day. So it's been great to use this as a tool to explain to people what I do and why I spend so much time doing it. So yeah, it's fun. It's great to have him here.
Are you have a brother? Do you have other siblings?
Yeah, another brother and a sister, and they're all over the place doing their respective areas of work, and yeah, it's nice to have one of them here.
Yeah, for that support.
Yeah, exactly.
Going backwards, where did you grow up?
I'm from Pittsburgh, Pennsylvania. Go Steelers. And then we moved around. My dad works in food and bev sales, so moved to Detroit and Atlanta. Ended up in the East Bay, Alamo. Anyone?
Anybody from Alamo here?
Yeah, my brother. So that's where my parents live now.
Yeah. What do your siblings do?
He works for Philadelphia Insurance. This is his first year out of Chico State, so that's what he does. And my sister's an actress in New York.
Wow.
Yep. And my older brother does some crazy tech stuff that's like monitors phones or for the CIA. I don't know what he does. It's all very hush-hush.
He's listening in right now.
Yeah, he can hear us. He doesn't need the cameras. Yeah.
Yeah, and your mom?
She's a stay-at-home mom.
Yeah.
Excellent mother. Works hard.
Yeah. So how many people are in your lab?
So I'm currently holding down as the only graduate student right now. We just had a bunch graduate, but we have a few postdoctoral researchers, some project scientists, and interns that come through CSU Channel Islands that come up to work with us, which is a cool program. And probably about 10 to 15 people, including the undergraduates.
So you have a line of 10 to 15 people doing the dance with the PPE at the end of the year.
Yeah, not everyone participates, but yeah, we get a couple of people.
How did you get interested in Grad Slam?
So at UCSB, it's been around for 5 years. And I remember when it first started, it was about the time I was going into graduate school. And actually, my roommate last year won UCSB's Grad Slam and came here, Nicole. So I saw her do it, and I was like, I think I could do that.
Right. So how was your campus competition? Tell us about that.
It was great. Our finals round was incredible. We had 3 rounds, so preliminaries all 1 week, and then a semifinals round and a finals round. And the talks were excellent. Just no one went over time. No one really stuttered. The finals were polished. I I was 5th or 6th in the lineup. I was like, oh, that was good, and that was
Yeah.
good. Oh gosh. So yeah, it was really great and really well organized as well.
Yeah, so you obviously prepared a lot for your presentation. Talk about that a little bit.
Well, I think there's a lot of people who also have my presentation memorized because I've given it to them so many times, and to my bathroom mirror. But yeah, it was a really big challenge to fit 4 years into 3 minutes. But I think it's a really useful exercise, as people were alluding to earlier, to be able to concisely explain what you do to get people excited about your research.
Right, right. So what's the most difficult thing in terms of translating research into language that a layperson can understand?
I think just coming up for synonyms for a lot of the jargon that we use every day that we forget. There's no reason for anyone to know those words outside of our field. So that was kind of a fun part of the challenge.
Yeah,
Yeah.
yeah. Well, good luck to you today.
Thanks.
Thank you very much.
Thank you.
All right, so now I'd like to welcome our second contestant to the stage. Give it up for Kelly Swanson from Berkeley.
Everything we see, the Earth, the planets, the stars, these make up only 5% of the universe. What is the other 95%? We don't actually know. Of the matter that we can see, what is it made out of? Are the smallest constituents tiny point-like particles? Or are they, as some people believe, vibrating strings? And when these particles move, what sort of dimensions do they move in? The Large Hadron Collider, or LHC, was a machine designed to answer such questions. It is the world's largest particle accelerator, spanning France and Switzerland. It is a ring inside which 2 beams of protons are accelerated and collide at nearly the speed of light. Out of this collision comes a whole slew of new particles that we can then study for signs of new physics. These machines have been instrumental in answering questions about our universe, but they are becoming prohibitively large and expensive. The LHC is 17 miles around and costs billions of dollars. We need a fundamental change in the way that these experiments are performed. And that is where I come in. My research aims to make such experiments more accessible by developing a tabletop-sized particle accelerator. In my lab, we take a laser with the power equivalent to a billion light bulbs and shoot it into a very thin thread of plasma. The force of this laser hitting the plasma creates a wave inside the plasma, and particles can then surf this wave to higher speeds. This is called laser plasma acceleration. This is one of our structures. It is only 3.5 inches in length, but can accelerate particles up to speeds that would require over 500 feet using LHC technology. But there are limitations to what can be achieved in this single structure. Any high-energy accelerator requires hundreds of these to be staged, such that the particles accelerated in one stage are injected and then further accelerated in another. Now, this is incredibly difficult as it requires exceptional control. The second stage must catch the particles from the first with a tolerance less than the width of a human hair, and everything must be timed to within a quadrillionth of a second. Now, as hard as this sounds, we have just recently demonstrated staging in 2 laser plasma accelerators, and the entire setup fits in the palm of my hand. Now, 2 laser plasma accelerators are not enough, but we believe that in the future, our research will really enable us to discover what the universe is made out of on a system that is small enough to fit on top of a table. Thank you.
All right. So what got you interested in making a tabletop of the Large Hadron Collider?
Yeah, so from a very early age, I was a big sci-fi reader. I'd read all these science fiction books, and I was like, oh man, I want to build these things. Right, so I built a really small particle detector in my basement when I was little. My parents were very thrilled.
How old were you when you were doing this in the basement?
Early high school. Yeah, so then I went to these particle accelerators. They have one outside of Chicago as well. So I went and visited, and they were huge. And I was like, man, I can't put this in my basement. So yeah, that's how I got into this field.
Wow. So tell us a little bit about yourself. Where did you grow up?
So I grew outside of Chicago.
So that's where the basement is.
Yeah.
Right. You still have a small collider in there?
No, not yet. But maybe I can take one home with me.
Yeah. So you grew up outside Chicago and ultimately made your way to Berkeley.
Yes.
Tell us about your journey.
So I went to undergrad at Caltech, which is outside of LA. And then I was like, oh man, the weather in California is so much better. I couldn't leave. So then I came up here to Berkeley for grad school.
Oh, that's great. And so I was asking contestant number one, how many hours a week she spent in her lab? How many hours a week do you spend in doing research?
I'm probably supposed to stay only like 40, but definitely more than that.
And what do you like to do when you're not doing research?
Yeah, I'm a big runner. I enjoy distance running, so ultra marathons take up most of the time that I'm not in lab.
So to relax, you run?
Yeah, it's hard to explain, but it's my happy place.
Do you race?
I do. I enjoy doing races. Yes, it's nice because they feed you along the way.
And so that's a way of getting food.
Yeah.
Yeah.
You know, I'm a poor graduate student. I have to find food where I can.
How do you, you said that you got interested in this field reading science fiction. So you obviously got interested at a young age.
Yeah, very young.
Did you always see yourself as doing research in a lab?
Yeah, pretty much. Well, in the beginning, I wanted to be a spacecraft pilot. You know, traveling between planets, but right now that's not possible, so yeah.
Yeah.
Always knew I wanted to be a physicist.
Yeah. Did you happen to see the movie The Martian?
I read the book, fantastic book. But I was like, the book is so great, I don't want to see the movie because then it would ruin it.
Yeah.
I like.
There's a great line in the movie where Matt Damon, who plays the guy who's stranded, has to figure out how to get his food supply. He's not a runner, and he just says, "We're gonna science the heck out of it," which I think is a great line. Have you had the opportunity to go to Switzerland to see CERN?
I did, yeah. I actually spent an internship there, which was a great experience. I got to learn a lot about accelerators, and then so when I started doing sort of plasma physics work, I was like, this is awesome. We can make them a lot smaller. More people could use them. I don't have to fly all the way to Switzerland.
Yeah, that would be good. And how do you like Berkeley?
Berkeley's great. Yeah, Caltech is very small, and Berkeley's very big, so it was a slight adjustment. But there are a lot of opportunities in Berkeley. The campus is beautiful. The weather is great.
Yeah. So you're big on weather, I can tell.
Yeah.
Does your family still live in Chicago?
My parents live in Chicago. My sister moved to Minnesota. Not sure why.
Not for the weather.
She didn't choose it for the weather.
Yeah. So you're one of two children.
Yeah.
Yeah. So other than running for food, what other interests do you have?
I enjoy traveling a lot. I think that's part of why I went to Switzerland, to go travel around. I spend a lot of my free time and a lot of my extra money traveling the world because I like seeing new things and meeting new people.
That's great. Well, I've enjoyed meeting you, so thank you very much.
Thank you so much.
Good luck. All right, so we're moving on to contestant number 3. Please welcome to the stage Sam Westreich from UC Davis.
$5 trillion stowaways hiding inside your body right now. I'm talking about the gut microbiome, the collection of more than 1,000 different species of microbes that live inside your intestinal tract. Now, it's only in the last couple of decades that we've realized that a balanced and stable microbiome doesn't just aid us with regularity. It also plays a vital role for nutrient absorption and for keeping our immune system in fit and fighting shape. But when this complex environment is disrupted, it can contribute to obesity. It can lead to the development of food allergies in young children, and it can even cause life-threatening autoimmune diseases. And worst of all, 1 in 3 adults will suffer from a microbiome disruption at some point during their lives. Now, to study this complex environment, most researchers today use an older approach called 16S, which we can think of as barcoding. It looks at the sequence of a single protein from each of these bacteria to give us a general sense of what's going on there. But aside from giving us no information at all about the activities of these bacteria, 16S is low resolution when it comes to identifying organisms. Relying on 16S is kind of like trying to navigate the streets of San Francisco with only a blurry satellite photograph as your guide. I helped pioneer a better method called metatranscriptomics. Now, I know that's a big word, and metatranscriptomics does mean big data. This is the sequencing of not just that barcode, but of all of the active genetic material from all of these microbes at once, and then using a supercomputer to sort out the results. But metatranscriptomics can tell us exactly which microbes are present in the gut microbiome at a much better resolution than with 16S. And if that was not already enough, metatranscriptomics is the only method that doesn't just tell us who's there, but what activities these microbes are performing for us. Metatranscriptomics is driving through San Francisco with both crystal clear maps and with turn-by-turn GPS directions. I built SAMHSA, a family of programs that handles all of the supercomputer analysis of this metatranscriptome data beginning to end. I'm putting this program to work looking at chronic diarrhea in rhesus macaque monkeys, a condition that has important implications for our own irritable bowel syndrome, or IBS. I'm also evaluating probiotics and their effects on humans, determining whether those pills at the supermarket are really worth the price that we pay. But most important of all, I have released SAMHSA, free and open source, and researchers at other universities across the country are moving away from outdated 16S and putting my program to work, analyzing their new metatranscriptome data. With metatranscriptomics as our path forward, and with SAMHSA as our roadmap, we will finally be able to fully understand the benefits of these 5 trillion stowaways that live inside of us. Thank you.
All right. All right, so what got you interested in studying the gut and diarrhea?
You know, it wasn't what I started off wanting to do as a kid. I wanted to build fast and light spaceships like all children, but
Did you build one in your basement like contestant number 2?
I tried. It did not go that fast. But what happened was I came into graduate school and I ended up in a programming lab. And I had done no programming when I started graduate school. I knew nothing about computers. And I learned how to do the basics of coding, and then I rotated through other labs, but I kept thinking back to how much I could get done if I was building computer programs. And so that drew me back in. And for the gut, and for metatranscriptomics, I was given some data and told, find a tool to analyze this. And so I went out and looked, and there were no tools. And so my next step was obviously, all right, I will build the first tool to analyze this type of data.
Of course.
Of course.
Yeah.
And here I am.
Yeah, so how long did it take you to come up with this?
So just to build the first version of this program took the first 2 years or so of my PhD career. Some of that was learning how to program. A lot of that was going back and fixing programming mistakes that I made. And since then, I have continued adapting and improving this program there's actually 2 versions out, where one version is open to the public and anyone can use it without having access to a supercomputer. And the other version is one that people can install on a supercomputer they have to get even more powerful results.
And have you done that?
I have.
Yeah.
And it yields incredible results. This yields a huge amount of data, which is always challenging. But from that, we can pull out whatever we're after.
Yeah.
And so I think it's the big data move of the future for microbiomes.
Right, for studying the stowaways in our digestive tracts.
That's right.
Yeah, yeah. So I've asked the other contestants, I'll ask you, what do you like to do when you're not doing research and coding?
So one of the advantages of research and coding is that I can connect to my computer remotely from any place I have internet access, which means I spend a lot of time in coffee shops sitting around. Because of this, I've become a bit of a coffee connoisseur, and I also do some fictional writing during my free time. It helps engage the other areas of my brain that aren't so active when I'm trying to write super dense computer code.
Yeah, so describe for us something that you've written.
So I like to write what-if stories. I like to tell, if we actually make progress on all this research and we get to the future and we have all sorts of technical innovations, where does that put us? Or conversely, if everything goes wrong, what sort of environment are we going to face? So it's a little bit perhaps beyond the scope of where my research will take me in the next couple of years, but it's hopefully where we will move or not move as a society.
Yeah, so let's hope we're not in the what if everything goes wrong
That's
category.
right.
Yeah, yeah.
Let's try to avoid that.
Right, so you said you're a coffee connoisseur. Any recommendations for us?
So I think it's important to just taste as many types as possible. A lot of people know that there's different types of beer, right? You have lagers, you have stouts, you have IPAs, and everyone has their own taste for that. With coffee, there are a lot of different flavors and varieties, and there are lots of different notes. And so it's great to explore, but the danger is that after you've had really good coffee, going back to your lab and drinking the stuff that comes out of the communal pot there is not nearly as enjoyable.
A little bit of sludge in there?
Yeah, right at the bottom.
Yeah, yeah. Where did you grow up?
So I grew up in Rochester, Minnesota, in the heartland of the United States. And I did my undergraduate work at the University of Minnesota in the Twin Cities, Minneapolis and St. Paul. And then I spent a year working for Habitat for Humanity, building houses in the area as I tried to figure out what exactly I wanted my next step to be.
Yeah.
And it was after I finished that year, I said, you know what? I'm done with snow. I want to go someplace warm and someplace that's really a leader in genetics. And so I chose to come out to California.
Right.
And chose UC Davis.
Right. And has it met your expectations?
I think it's exceeded my expectations. It is amazing to not have to go out and scrape an inch of ice off of the windshield of my car every morning.
Yeah, there's a technique to that, you know.
There is.
Yeah.
It does help build the muscles, but you have to plan an extra 20 minutes at the start of your day for it, so
Right.
trade-off.
Right. So when you came out to Davis, you said you were interested in genetics. What got you triggered onto coding and directly into this project?
So that is a little bit of a story where I eat some crow. I came in and I was looking for a lab to join. And one of the people who interviewed me was one of my professors now, who's this expert in coding. And I went to him because I remembered the interview and I said, I like talking to you. I don't want to program. I don't want to do this for the rest of my life. I just need a first round slot to hold me over for a couple weeks. And he said, sure, yeah, come on in. I don't know why I said that, but he did. And I did that rotation, and I loved the programming so much that I had to go crawling back a couple months later, hat in hand, and say, I know I put down programming before, but this is what I want to do. Will you forgive me for mocking it at the beginning? And he just smiled and said, of course. And that was what brought me in.
Right.
So
Right.
try new things. You never know which ones you'll end up liking or making a career out of doing.
Yeah, absolutely. And tell us something about yourself that maybe your graduate advisor doesn't know.
So my graduate advisor knows a good amount about me, but I would say one of the things she probably doesn't understand is that I do some work from home because I can connect remotely. Most of the time, if there is a typo in my emails, it's because my cat has wandered across the keyboard.
Oh, you're a cat person.
I have one cat, and he is a spoiled lump whose microbiome I have not studied yet, but that is on my to-do list.
That was my next question.
At some point, hopefully I can graduate, I can get a job, and then I will have the time to start probing my cat and finding out more. Nope. I will have the time to start investigating my cat and learning all about the microbes that make him tick.
You know, I would be afraid to be your cat.
He's very spoiled. He's very lucky.
What's his name?
His name is Leo, but he's also known as Goober, because you have to give a cat a diminutive little nickname to make him not seem quite so smug and stuck up.
And in control.
That's right. It's
Yeah,
tough.
Yeah. So where do you see your future going?
So I think with the microbiome area, it's a very new area. I said it's only in the last couple decades that we've even started looking at it. And a lot of what's out there now is first impressions, and we have barely scratched the surface. I would love to keep working on this and to keep building the programs to dig deeper, but then also turn some of this into possibly a company or some way to put out this type of information and make sure that all these people who suffer from microbiome-related illnesses or troubles are able to make use of it.
Yeah.
Right now, it's still in its infancy. It's still in its early stages, and hopefully we can grow that and make it a lot more accessible to people, understandable to people, and useful for them.
Right, right. And your presentation said you were evaluating probiotics. Tell us about how you're doing that.
So for this study, we actually took a group of healthy adults and we've given them both placebos and probiotics over the course of several weeks, and we monitored how their gut microbiome changes. And what we found is that most probiotics don't do a lot for people who are already fairly stable. Your microbiome actually stabilizes quite quickly. It might not stabilize in the way that you want it, but once it is set, it's kind of like concrete, and you can't budge it with anything short of a sledgehammer, like antibiotics. But where probiotics could be more useful is for people who maybe they just finished an antibiotic regimen. They just had food poisoning or something that caused this disruption. And if we can pick the right probiotics that perform the right functions for these people, we might be able to help rebuild and restabilize their microbiomes without having to go through the guesswork and the long-term impacts that we face now.
Right, right. So that would come in handy.
I hope so.
Yeah.
For a lot of people.
Yeah, so you see yourself possibly going into the private sector versus remaining in academia.
I think so. I also, given that I'm standing up here, I love explaining and talking about this, so I think I would probably do pretty well as a teacher as well.
Yeah.
I want to do whatever lets me help the most people.
Yeah, yeah. So tell us about your growing up, and are you an only child, or you have siblings? Or?
I have one younger sister. When I grew up, she's 3 years younger than me, and we fought like wildcats. Now, she apparently
there's the cat theme again.
There's the cat theme. Just keeps following me. Now, apparently, though, she's following after my footsteps. She's actually starting at University of Washington, Seattle on her PhD in plant biology and insect pollinators.
Wow.
So she saw me go into grad school and for some reason said, yes, I also want to do that.
Wow, wow. How did your parents do this for their children? What did you guys talk about at the dinner table?
Well, my mother is a nephrologist. She studies kidneys, and so I grew up learning how to eat my creamed corn while she talked about the patients that she saw and how they had hypertension and liver problems and all sorts of things wrong with them. So I developed the iron stomach that maybe is needed for microbiomes from that. But they have always pushed science. My mother's a doctor, my father's an engineer, and they've always just encouraged me to ask why for everything.
That's great.
I think that got me here.
That got you here, and we're glad you're here. Thank you.
Thank you so much.
Congratulations.
Thank you.
So now it's time for contestant number 4. Give it up for Byron Furman from UC Merced.
So just a quick show of hands, how many of you drink water or something with water in it in the past 24 hours? Great, thanks everyone. You know, water is a really important part of everyday life, but we tend to take it for granted. I mean, I'm a part of the water industry myself, and I don't even think about it when I go walk over to the kitchen and go grab a glass of water. Wait, where is this water even coming from? Well, worldwide, there's generally 2 ways that people get drinking water. In industrialized countries, we have wastewater treatment plants, which are extremely expensive. They take a ton of energy too, but they have a huge output to meet our kind of ridiculous demand. In developing countries that don't have the same number of resources, they rely a lot more on natural systems, where you let the water flow through environments that pull out the contaminants along the way, so you have a much cleaner output at the end. Now this is really important because more than 50% of the people on this planet drink untreated water. So they're drinking right from a local lake or stream or river nearby. And these water bodies are very susceptible to a problem called eutrophication, which can occur if there's a lack of oxygen in the bottom of the water, and it causes this biochemical reaction which allows algae to form a thick green sludge layer at the top. And this sludge will block out any incoming oxygen, so it'll kill any fish inside, and then it also releases toxins down into the water, so it's really just horrible for water quality. In a world where the population is already 7 billion people and it's rapidly increasing, a lot of these water security issues require cutting-edge technology. Honestly though, this isn't one of them. The solution is really just as simple as an aquarium aerator, which can get oxygen down to the bottom of a fish tank, right? Okay, so obviously we need to scale this up, but the concept is still the same because all you really need is a pump, some tubing, and then a diffusion system to make it mix at the bottom. My research team is mostly involved in preliminary evaluation of these water bodies and system implementation. So you might find me out on a boat collecting water samples, or you could even see me in the lab monitoring chamber experiments, where I can test how a water body is going to react to this type of system. An example of one of these chambers is just over on your right. And so in a world where we have so many of these water security issues, we really need to look for this solution. And my research team has found that this results in dramatic improvements in water quality, and it's thousands of times cheaper than wastewater treatment options. I also have a background in business, but I really don't think you need a business degree to know that money talks, right? If I approach an official in an area that suffers from this problem and say, you have $100 million, you have a problem, but I have a $100 million solution for you, what do you think? He probably won't even let me finish explaining myself. But if I say that the same problem has a simple and affordable solution, he's much more likely to take me seriously. This system has the power to protect fisheries in areas that depend on them as a staple. It can make sure that people that can't just walk over to the kitchen to go grab their water, that they don't have to partition this thick green sludge layer off the top of the water and dump a bucket in for drinking water. Essentially, it brings improved water quality to the places that need it the most. Thank you so much.
All right, now we've done our spontaneous photo. So what got you interested in studying water?
It's been a really long journey, to be honest. I started off in chemistry, and I wasn't really quite sure where my field was. And so I started working for an environmental laboratory, and it kind of got me thinking that the big picture is really looking at water quality, especially where I was working. I got to see a lot of environmental engineers that were always out in the field monitoring water bodies and kind of making sure that the water quality is good around here. So I wanted to bring that around the world, not just keep it here.
Yeah, so have you had the opportunity to travel to some of the countries that have problems with their water quality?
I honestly haven't yet. This is kind of a newer thing that's been developing the past few years or so, as long as in my line of research, but it's been really rewarding. I actually just started at UC Merced. This is my first year.
Yeah.
But my advisor, he is amazing. He just knows so much about these types of systems.
Well, Merced has a great faculty, and they do some great research there. Where were you before you were at Merced? Give us your life story.
Oh wow, that's a long one, even though I'm kind of young. So I started out in Anaheim. I went to Humboldt State University for my undergrad, and then I had to stick around for a little bit longer and get an MBA too, so I could figure out what I wanted to do. And then that was while I was working at the environmental lab. And so after that, I went on to University of Florida for some environmental engineering. And then that brought me all the way to UC Merced.
Oh, that's great. And you're enjoying yourself at Merced?
Oh yeah, I love it at Merced. It's just such a great place to grow. Everything is still developing, and it's wonderful. Like if you saw the buildings, they're all 21st century. Everything is really carbon neutral. There's a huge push for sustainability, and it's kind of a place where we can find ourselves. Like it's not ultra established and everything is black and white and you have to do exactly this and that and this.
Yeah.
There's so much flexibility that I love just working there and being able to kind of create our own lab
Yeah.
rather than having the lab that's already like it is.
Yeah, one of the advantages of going to our newest campus, Merced. And what do you like to do when you're not doing research on water?
It's a little bit different, but I really love martial arts and boxing.
Really?
That really just gets my blood flowing. I'm an adrenaline junkie. I like skateboarding too, so it's kind of I don't know, I guess hurting myself a little bit.
What kind of martial arts do you do?
I grew up doing Taekwondo. I absolutely love it. And then boxing was my second favorite.
Wow. So do you box competitively?
A little bit. I'll do some low-key amateur stuff, but
Yeah.
you know,
I've always-
research is my future, so.
Yeah. Yeah.
Got to take care of the brain.
Not boxing. Right?
Exactly.
Right. I've always wondered watching boxing. I mean, does it hurt when you get punched?
I think everyone's different. Some people you can see that they really just don't like being punched. To me, I'm not gonna lie, it's a little bit of that adrenaline rush of like, oh, you hit me, that didn't even hurt that bad. Like, come on, bring it more. But you know, everyone's different.
Yeah, I guess so. I think I think most people don't like pain, really.
Yeah, I think I'm a little weird like that. But.
Yeah. Are you an only child? Do you have brothers and sisters?
No, I've got a sister. She's brilliant. She got a degree in electrical engineering from Texas Tech University.
Wow, she's a Red Raider.
Yep.
Yeah.
Yeah, my dad is as well. They were a little disappointed that I had to stay in California.
Tell us something about yourself that maybe your graduate advisor doesn't know.
He probably doesn't know how I'm going to incorporate my MBA into my work, but I think I got the master plan all up here. It'll
Yeah?
work out in 10, 15 years.
Yeah. So what is the master plan?
I would be lying to you if I didn't say that it was this project and bringing it worldwide and really just trying to improve water quality and say, you know, who has the worst water quality in the world and how can we do even something small just to improve that and go from there.
Yeah.
I think that we can make some really huge impacts.
Right. So particularly in the developing world, right?
Yeah, there's a lot of problems with eutrophication in Asia, Africa, and South America. Not that I've seen any of it myself because I haven't traveled there yet, but that's definitely part of the goal.
Yeah, to get on the road and maybe even market your. Is it a device? Is it a yeah? Can you describe it?
Somewhat, yeah. It's basically a big fish tank air pump. We use them a lot in the U.S. because they're really great for water quality. So when a lot of these lakes and reservoirs are drinking water reservoirs, so they just hold our drinking water. So when they pump that out to the wastewater treatment plant, it's just so much easier to treat it and get it to the people. So with people that don't have that treatment option, they don't have the millions of dollars of capital to have a real wastewater treatment plant in operation. I think that the second best option is really to just take away the problems of eutrophication, make sure that the people aren't drinking green water,
Yeah.
and then we can move on to getting them even cleaner water afterwards.
Right. So you're basically inserting, as I understand it, you're basically inserting oxygen into the water. Is that what's happening, or?
Exactly. So the concept works the same with oxygen or air. The air is about 20% oxygen. So if you want to just keep it from eutrophying, you can use normal air. And then if you want to give it an extra boost of oxygen, then you can use liquid oxygen.
Yeah. Give it a little jolt.
Exactly.
Yeah. How do you explain what you're doing, say, to your parents?
Let's see. So they're actually, my dad's really intelligent. My mom, I got a little bit of her personality. I love to talk. She's a big talker. And yeah, I mean, with my mom, we could just sit down and 4 hours later, we would have talked about everything under the sky. And my dad, he's just so quick that he catches on right away. He knows what I'm doing, even though he's totally opposite field. He works in HVAC or heating and air conditioning.
Yeah. What got you interested in doing Grad Slam?
It was almost a whim. I was almost just like, I want to get better at presenting. I kind of enjoy this, but I'm sure I could learn a lot more, get some tips and tricks from people. And as I went through, I started realizing just how passionate I am about my research and how, the more that I improved my speech, the more that I really liked what I was doing, and the more that I could tell it with enthusiasm. And it's been a great experience.
Yeah.
I would recommend it to everybody, no matter what your position is in grad school. If you just started like me, give it a shot. You have nothing to lose.
All right, well, with that, we'll say thank you.
Cool. Well, thank you so much.
Give it a shot. All right. All right, I'd like to welcome to the stage contestant number 5, John Feltz from UC Santa Cruz.
Good morning. Imagine you've driven out to the coast. It's a warm, sunny day. You're relaxing, swimming in the ocean. All your worries disappear. But then as you're sitting there, pieces of trash start floating by, and your liquid dream quickly turns into a plastic nightmare. I'm a surfer. I imagine a few of you here might be as well, and as surfers, we care deeply about the ocean and the environment. However, the equipment we ride couldn't be more toxic. This is an art installation called the 1000 Surfboard Graveyard, and its goal is to bring awareness to the carbon footprint and waste issues associated with surfboards. Now, a surfboard's biggest contributor to waste is the foam core called the blank. These foam blanks are typically made using the polymers polyurethane or polystyrene. The issue with these foams is that they're non-renewable, they're toxic, and non-biodegradable. Polystyrene foam, or as you might know it's styrofoam, takes over 500 years to degrade. And that's why right now in the US, our landfills are filled nearly 1/3 by a single material, polystyrene. Surely there must be a better material to make these foam blanks out of that is sustainable, that is non-toxic. Even better, what if this material is something that's currently just going to waste? Over 8 million tons of shrimp, crab, and lobster shell waste are disposed of globally every year. Of that 8 million, over 2 million of it is a biopolymer called chitin. But what is chitin? Chitin is a type of natural polymer called a polysaccharide, and it has a chemical structure very similar to that of cellulose. So if you think of cellulose as the tiny fibers that make up trees, well, chitin is the tiny fibers that make up shrimp shells. Chitin is the second most abundant polysaccharide on the planet after cellulose. It's also non-toxic, biodegradable, and sustainable. So with the enormous amount of chitin disposed of every year, it'd be amazing if we could turn this otherwise wasted material into foam and start replacing these toxic polyurethane and polystyrenes. Cruise foam does exactly that, taking chitin from shrimp shells and transforming it into a foam. Our eco-friendly, water-based process involves only water, salts, and a foaming agent. There are no toxic components. Our proof of concept seen here has comparable mechanical properties to that of existing polyurethane and polystyrene foams used in surfboards today. The application opportunities for cruise foam are immense. With further research and development, it has the potential to go far and beyond surfboards. Thank you.
Right. All right, John, so were you always interested in making surfboards out of shrimp?
No, definitely not. My bachelor degree was actually at UC Santa Barbara, and that's where I did chemical engineering. And so I've always been interested in chemistry and engineering, and it wasn't until I actually went up to University of Washington and got my master's in materials that I really got a lot more entrenched and a lot more excited about material science. And coming down here, I've been surfing for about 12 years. It was kind of a no-brainer. I've always known the issues with it, and I kind of put all my background together. Being in Santa Cruz, it fit perfectly.
Right. The surfing, the Santa Cruz, and the shrimp.
Exactly.
Right.
Exactly.
Right. So Did you always know that you wanted to go into material science, or how did you find your way there?
Yeah, like I said, I did my bachelor's in chemical engineering, and I actually went into industry for about 5 years doing environmental remediation on Air Force bases at Cape Canaveral for a consulting company. And I enjoyed it. I learned a lot. I got to see a lot of cool things. I got to see the second to last space shuttle launch, so that was pretty amazing. But after doing that for about 5 years, I don't want to say I got burnt out, but I definitely wanted to try something else. And I wanted to go into engineering. I wanted to go back to school. I didn't quite want to go back to chemical. And materials kind of just, it spoke to me. It seemed like the right fit. It kind of seemed to where a lot of jobs are going. And it was the right choice.
Yeah, yeah. So, but let's talk about surfing.
Yes, please.
How long have you been surfing? How often do you surf? You know, we want to know your surfing career.
No, of course. No, I learned how to surf in Santa Barbara. That was a while ago. Like I said, almost 12 years. So I've been surfing for some time. I surf, if I can, once or twice a week. You know, the lab keeps me pretty busy. Unfortunately, I also had my appendix removed 2 weeks ago, unexpectedly. So I haven't been able to surf. And it's been killing me, so.
Well, so will your appendix if you don't.
Yeah, well, at least I don't have it, so.
Yeah. So, what else do you like to do in your spare time?
I am a very, very large Arsenal soccer supporter. It's an English Premier League team.
Yeah, I heard of them.
It's a rough year for us, but I've been supporting them for almost as long as I've been surfing.
Yeah.
So that is something I'm extremely passionate about.
Yeah. How did you get?
Yeah, it's obviously it's on the other side of the world. One of my roommates, my freshman year at college at Santa Barbara, is actually from El Salvador, and he was a big Liverpool fan. How he got involved, I'm not really sure, but he's a big Liverpool fan, and I saw him watching the games. I wanted to watch him with him, and I couldn't pick the same team as him because that'd be no fun. And I didn't like to go for the big name teams like Manchester United. And at the time, Arsenal had some really great players, Thierry Henry, Dennis Bergkamp. The team sometimes chooses you, and I've watched them, and I've loved them ever since.
Yeah. So. Yeah. So, do you watch them online, or how do you?
It's rough. Luckily, the coverage here is pretty good. There's a NBC Sports actually gets more coverage than the people in England, which is hilarious. But the only issue with that is sometimes I'm waking up at 4 a.m. to watch some of these games because they're way on the other side of the world. So
you are dedicated.
I am dedicated.
Maybe insane, but
yeah. It works for surfing too because it's about the right time. The game's over. It's dawn outside, or the sun's rising. Time to go surf.
Wow.
So
wow.
That works.
Sounds like a good day.
It is a good day when it works out like that, and Arsenal wins. It's a good day.
Yeah. So tell us something about yourself that maybe your graduate advisor doesn't know.
It's Arsenal related. Sometimes those games, they'll have, there's a couple competitions and they happen to be around lunchtime. So I will take a long lunch, and he might wonder where I am sometimes, but he knows I'm a pretty big Arsenal fan, so I don't think he's too unaware of what might be happening.
Yeah. So where'd you grow up?
I grew up actually in the East Bay. I grew up in Pleasanton. Grew up there, and I lived there obviously, and then my parents moved down to Pismo Beach when I graduated to go to Santa Barbara, but I love the Bay Area. I've spent the majority of my life here. It's a great place.
Yeah. Where do you see your future taking you?
Ideally, this will take me into a company. We actually, me and my advisor, we actually recently filed the paperwork and had this incorporated. So we are an official company at the moment, but a lot of work still to be done, obviously, not only the research, but moving forward. But that's ideally, this would be my future.
All right. Well, good luck.
Thank you very much.
Thank you. Congratulations.
Thank you.
It's now time for our sixth contestant. Please join me in welcoming to the stage Christina Heschen from UCSF.
Say you want to build an elephant from scratch. from just one cell. Imagine how many times that cell has to duplicate and divide to get from a one-cell embryo to an enormous animal. By my rough calculations, divide by 2, carry the 1, 200 million cells divide per second in the body of a growing elephant. So the question of how to build an elephant is actually a question of how a cell divides itself from one cell into 2. To divide, cells build a super reliable machine called a spindle, here in yellow, that literally grabs your DNA, lines it up, and then pulls half the DNA into one new cell and half into the other. If the spindle loses grip of any DNA, new cells won't have the full DNA blueprint that they need to survive. Even a small mistake can lead to developmental disabilities like Down syndrome or to cancer. In other words, you should all care a lot about spindles, because without spindles, there would be no cell division, so there would be no elephants, or, humans. For my thesis research, I wanted to figure out what makes spindle machines so reliable. And to understand a machine, you have to take it apart. So, one day, I shot a laser into a cell to cut the spindle, setting loose precious DNA, and then watched through a microscope to see how it reacted. Amazingly, that laser-cut spindle rebuilt itself within 30 seconds. This stunning observation raised 2 questions. 1, how does the spindle sense damage? And 2, how does it repair itself? Eventually, I figured out that one of the 2 fresh cable ends created by my cut, the one still attached to the lost DNA, serves as the alarm that alerts the spindle to danger. Then, the spindle sends out a repair team specifically to this end. Long, sticky molecules are the first to recognize the cable end alarm and grab hold of it. They pull in an army of tiny, 2-legged engines that drag the lost cable and DNA back into the spindle, guiding it home. This built-in alarm plus responder system allows the spindle to detect and fix structural damage, keeping hold of all our precious DNA. What I find most amazing here is that the spindle is a self-repairing material. Imagine if roads or buildings could just fix themselves up after an earthquake. Right now, inside each of us, 2 million spindles are doing just that, this dynamic dance, rebuilding and repairing themselves to ensure that they never make a disease-causing mistake. That reliability is what builds an elephant. Thank you.
All right. All right. So what got you interested in studying cells and spindles and elephants?
Yeah, I'll take each of those one at a time. So I actually started grad school having done some research in sort of straight classical cell biology, and that's what I thought I wanted to do. But I ended up doing a rotation my first year in Sophie Dumont's lab, which I ended up joining, which is a neat place. We're kind of a young lab, and we have some people with cell biology backgrounds, but also some people with physics backgrounds. And we study, as you heard, mechanics and forces involved in cell division. And that kind of hooked me, that interdisciplinary space. And so I got excited about these kinds of physical questions in cell biology. And the spindle, right, has to do this very physical task of moving giant chromosomes through the dense cell on huge distances that are many times larger than the distance or the scale of individual molecules. So a lot of people who are interested in these mechanical questions in cell biology use the spindle as a system to study them, and that's how I got interested in spindles specifically, which are amazing.
Right, and then the elephant question?
The elephant, right. Actually, someone asked me why I picked an elephant the other day. I think it was just because it's so large. Like, it does blow my mind to imagine that one cell turned into those billions of cells. But I don't know, I've always liked elephants. I have seen some of them in person, and those were great experiences, so maybe
They
that
seem like
tied
nice
into it.
animals, yeah.
Yeah.
Yeah. So tell us about your journey. How did you get to UCSF? What's your history here?
So I'm from the Bay Area. I grew up on the peninsula. And let's see, before grad school, I was working for a couple years abroad and then in DC in a lab at NIH. And when I was applying to grad school, so I decided I definitely wanted to keep doing research, wanted to go to grad school. And I knew it would be a pretty long haul, so I think I wasn't sure about committing those 5 or 6 years to anywhere else in the country and decided to come back to the Bay Area. And UCSF has a reputation for being a super collaborative environment. I think it really deserves that. That's something I've enjoyed a lot, so it's been a good fit.
It's been a good fit. What do you like to
Yeah.
do in your spare time?
Yeah, so I actually recently have taken up surfing a little bit. But
So
I'm
you're interested in the
terrible.
shrimp shell thing.
So I use this huge decades-old foam board that I now feel guilty about. Let's see, what else? I spend a lot of time on the weekends, I would say, in a couple of favorite bakeries, eating baked goods. I don't know if that counts as a hobby, but I do a lot of that.
Does
Also
for
like
me.
to dance. Yeah.
You got some recommendations?
Yeah, if you're ever over in the inner Sunset, we have a bakery called Arizmendi that's incredible and well-loved. I see some nods. And I also like a couple of the Chinese bakeries there, or Shenki, which is a chain. That's a favorite of mine.
Oh, you're making me hungry. Are you an only child? Do you have siblings? Tell us about growing up.
I have one younger brother who's sitting over there, actually.
Another younger brother in the audience.
He's great. He's 4 years younger, so
Yeah.
I think we didn't fight too much growing up. We've always been pretty
No
close.
boxing?
No, I mean, maybe very early on. Mostly initiated by him, I would say. I'm going to put the blame there.
Tell us about how much time you spend on average in your lab versus surfing or eating baked goods.
Yeah, their comparison might be more fair there. I don't know about something. I've never really tried to calculate that either. Some of our work, too, we do a lot of quantitative image analysis, and so some of that can be done at home. Sometimes on the weekends, I'll do a little extra of that. That might be where most extra hours come from.
Yeah, yeah. And you said you started off thinking about cell biology, but did you always want to be a cell biologist? Let's say when you were in the 4th grade, did you say, I want to be a cell biologist?
My parents are also here. They might be able to answer this better than me. But I think actually my first love might have been stories and storytelling. At least I remember always getting in trouble for like reading novels when I was supposed to be doing other things.
Like homework?
Yeah, or like at night after I was supposed to be asleep with a flashlight or something. But I think I had some scientific leanings. Like I remember maybe some proto experiments, like thinking about what dirt would taste like and then tasting it to find out, or something like that. So maybe there were two.
Yeah,
poles.
and what does it taste like? We want to know.
I actually remember that pretty, I don't know, sort of like when you get a baked good that's just really chalky, or like, I'm back to baked goods, clearly. I don't know.
Yeah,
Or
I'm
like
getting that
really
theme.
bad chocolate soft serve when it's done really terribly and it just tastes like powdery chalk.
Like gunk.
Yeah.
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 Yeah. So did you have a favorite book that you read as a child? Does anything stick out in your mind?
Very favorite. I mean, as a young child, I was very into the Hardy Boys and Nancy Drew series. I read like every copy that our library system had and the next town overs.
Nancy Drew
That's a good one.
and Ned and his little red roadster.
Exactly.
And,
But I
yeah.
actually like the Hardy Boys series better. I think it's a shame they're so gender marketed. Everyone should read the Hardy Boys.
Where do you see your future going? What is your pathway, Christina?
Yeah. So I'll probably graduate in about a year, and I've been thinking about this a lot. I do really enjoy communication, and that's something I'm interested in, as well as education. They're kind of tied together. But right now I'm thinking that staying in research and hopefully academic research kind of combines the research interests and those other interests best, or I would like to use it to combine them if possible.
And to possibly teach, or?
Yeah, ideally. I think this event and this whole process has, I don't know, been a good reminder of how we can and should combine all those things.
Yeah. So how did you get interested in Grad Slam?
I saw it last year, and it was, let's see, and I had some friends who participated last year and who said they just enjoyed the process, which I definitely did too. Yeah, so I guess I just thought it would be both fun and kind of a good skill building exercise.
Yeah. So what was your campus competition like?
It was great. It was really well organized.
Yeah.
I mean, as is.
And
this
you
one.
did well.
Yeah, I guess so. But yeah, it was, all the talks were amazing. It was a lot of fun.
Yeah. Yeah. So how much time went into your preparation for today?
A fair amount. I think more before the UCSF competition, and I didn't change it too much for this one. But I did a lot of practices on non-science friends, or a couple of friends who were design majors were actually really helpful. Like, I did it for them, and they looked at my slides and told me what things they thought the slides were highlighting and what not. So I think incorporating feedback from a lot of diverse audiences maybe took the most time.
And did you focus on the elephant initially, or was that added in
The
as
elephant was
a
always
dramatic
there.
Ending, you know?
Actually, I had used the elephant, not to the same degree, but I'd used the elephant in some talks for my graduate program, or for sort of slightly broader audiences that were still scientific. So it wasn't, that was actually where I started from, I
You
would say.
started with the elephant.
Yeah,
Yeah.
good place to start.
Because you like elephants.
Right.
Yeah.
Right.
Yeah. Yeah. Do you have any
doesn't?
pets?
We had dogs growing up. My parents still have a dog, but I currently do not. I'm actually not allowed any pets at my apartment.
So that's too bad. No pet elephants.
No. Yeah.
Yeah.
That
That
would be ideal.
That would be difficult in a San
would
Francisco
also be
apartment.
difficult.
Yeah. Yeah. How big is the lab that you work in?
It's pretty small, but growing. I think we're up to 8. It's been a really fun environment. We're a young lab, as I said, and everybody's very enthusiastic, and it's a great, fun environment.
Yeah. Yeah. Tell us something about yourself that maybe your graduate advisor doesn't
I
know.
was thinking about this while you were asking other contestants. I think there's not a lot that she doesn't know. We're a pretty close-knit group. She might not know how much I like to dance and how much time I spend per week at UCSF's various dancer-sized gym classes, which are a favorite pastime of mine.
So what else do you like to do in your spare time? You said bakeries, you said surfing, now we've got
dancing,
dancing. What else you got going on?
I like to run, although I wouldn't say I'm a runner, nothing like Kelly.
Yeah.
But
you don't run for food.
No. Yeah. Well, actually, I often end my runs with some sort of snack or baked good before I walk back to my apartment, so maybe it counts, but I pay for it in that case, unfortunately.
Well, that sounds like a great plan. Congratulations.
Thank you.
Okay, we're moving forward to contestant number 7, Nathan Harrell from UC Riverside.
Sense of self and sense of self in relation to others comes from how we are treated as children by our parents. We learn who we are in relationships, and we learn what intimacy is through the relationship with our parents. For example, if someone gets physically abused throughout childhood, they may grow up and find themselves in abusive, chaotic relationships because that's the intimacy that was taught to them. If someone gets neglected throughout childhood, maybe they grow up to find themselves attracted to emotionally unavailable people, like their parents were emotionally unavailable. So people who are much older or married or otherwise off limits and not really present in the relationship. Sometimes people who are neglected will develop personality features to capture the attention and respect they didn't get when they were kids. One way this can manifest is they become academics. And now they're so smart and everybody's got to listen to them. So the point I want to make with these examples is how you're treated as a child can influence who you're attracted to, who you marry, your career, all sorts of things about your personality and how you relate to other people. So given that parental care is so important, what do we understand about the neuroscience of it? Well, it turns out there's a small cluster of cells in a brain region called the hypothalamus there, where if you lesion or kill these cells in birds, in fish, in mammals, we are mammals, the animal won't behave parentally much anymore. Or if you activate or stimulate these cells, the animal will behave parentally towards any baby you put in front of it. So that's really interesting. What's going on here? That's what I figure out. I measure all sorts of properties of these cells. I figure out their shape. I figure out how they're connected to other parts of the brain. I figure out what they hear when other cells talk to them. I figure out what they say when they talk to other cells. I measure all sorts of properties. And I do this in a rodent, a mammal, where the virgins are abusive. If you put a little baby in front of them, they're going to ignore it or attack it. That's neglect or physical abuse. Now the parents are great. They love babies. You put a baby in front of them, they'll run up to it, make sure it's warm, take it back to a nest they built for it. They're wonderful. If you take a virgin and you inject it full of hormones, the same hormones that change in you when you become a parent, now all of a sudden they love babies. That's because the hormones go up into the brain and they change the properties of cells that I measure. So if you want to understand why you love your children, if you want to understand why people abuse children, you have to fund research like this. Ultimately, we want to be able to change the brains of people who are wired up to abuse children more efficiently than we do now. And in order to do that, we got to do some basic research. Thank you.
Congratulations.
Thank you.
All right, Nathan, so what got you interested in this topic?
Well, I kind of stumbled into it, but if I had to say two main things, one, when I was a teenager, I'd listened to this radio show called Loveline. I don't know if you all heard of that. With Dr. Drew Pinsky and Adam Carolla, believe it or not, kind of a raunchy comedian. But it's like a national radio show. People call in, they talk about their relationship issues or parents, and they give advice over the phone. And that's where I started cluing in on this really interesting stuff about parent-child interactions. In college, I went to a small liberal arts college where there was Dr. Melissa Burns-Casado was doing some research on parental care in doves, and I stumbled into that lab, and then that got my foot in the door for a parental care lab in graduate school.
So you're studying it in rats now?
I study in a kind of a rare species called California mice, Paramyscus californicus. I focus on paternal behavior. Like around 5% of mammals engage in paternal behavior. The other 95% is just moms doing the parental care.
Yeah, we know that, yeah.
I kind of focus on paternal behavior in this species, yeah.
Yeah. So where do you think your research will end up? What will it give to us?
Yeah, it's hard to imagine what the future will hold. You know, a long time ago we had no idea about CRISPR, Cas9. So the more and more we figure out about the basic neurobiology of parental care, the more and more we're going to be able to correct people with negative child interactions. So it's hard to imagine what that's going to be like, but hopefully improve screening and treatment for child abusers.
Yeah, that would be awesome. What do you do when you're not doing research?
In my free time, I like to try to graduate. No, I'm just kidding. I like to exercise, go to the gym. I got a surfboard too.
So there you go. Another shrimper. Yeah. Have you been surfing a long time?
No, first year when I came out, there was an older graduate student that was into surfing, and so I was raised in Kentucky. I never thought I'd end up on a coast surfing, but he got me into it, and it's a great way to blow off steam and relax.
Yeah, yeah. I've been asking people about their families. Do you have siblings?
Yeah, I got one older brother on the East Coast. He's a fancy lawyer doing intellectual property stuff.
Fancy lawyer.
Yeah.
Yeah, those words often go together. Yeah. Tell us something about yourself that your graduate advisor doesn't know.
My grad advisor doesn't know that I work every night and every weekend. Hi, Wendy. She's probably watching me. I'm not sure. We're pretty close. Yeah, I don't know.
Yeah. So he knows or she knows everything.
Not everything. She doesn't know both my parents are twins. That's probably a fun fact she doesn't know about me.
Okay. We'll write that one down.
Sure.
Yeah. What did you want to be when you were growing up? When you were in the 4th grade or whatever, did you think you would be studying the neurobiology of the brain and its effect on behavior?
No, I don't even know if I had that much foresight to want a certain career when I was tiny. I don't recall anything. I just kind of stumbled into neuroscience in college. My small liberal arts college had a behavioral neuroscience major, which I guess is kind of rare. And so that was a great opportunity to get interested in this stuff.
Yeah. So you got interested as an undergraduate, and then
Yeah.
and did you go right from undergrad to grad school?
Yes, straight out that gate from undergrad.
Right. So you say you spend your spare time trying to graduate. You got a timeline on that?
Hopefully in a year or two, maybe closer to a year if everything goes well. Fingers crossed. Yeah, but in a year or two.
All right. Well, good luck to you.
Thank you so much.
All right, please join me in welcoming to the stage our 8th contestant, Megan Newcomb from UC Irvine.
Global warming is caused by increasing levels of carbon dioxide, or CO2, in our atmosphere. A few people may disagree, but the facts don't lie. This greenhouse gas is increasing global temperatures. Other gases, like the pollutant carbon monoxide, or CO, contribute to the formation of smog that is plaguing cities around the world. CO and CO2 are also byproducts of the fossil fuels we depend on. This is a big problem. But what if I told you that nature has already developed the machinery we need to solve it? I'm talking about enzymes. Enzymes are tiny little biological machines. All forms of life have them, and they do everything for us. They chop up our food, make our DNA, turn light signals into visual images, everything. The really cool thing about enzymes is that they can do very difficult chemical reactions at normal temperatures and pressures. In the Ribby lab at UC Irvine, I study an enzyme called nitrogenase, which is found in some types of bacteria. Nitrogenase can take CO and CO2 out of the atmosphere and turn them into propane and butane, which humans use regularly as fuels. If scientists wanted to do the same thing without the enzyme nitrogenase, they would need high temperatures, extreme pressures, and expensive rare metals. My research focuses on understanding how this enzyme does these difficult chemical reactions at room temperature and normal pressures. Like the machines we are used to working with, nitrogenase needs many parts to function. In my research, I take nitrogenase apart and put the pieces back together in different ways to better understand how it works. One of its important parts is a big group or cluster of metal atoms. We know that these metal clusters power the enzyme, but my research shows that they play an even bigger role than we originally thought. By further understanding which parts are the most important to nitrogenase's activity, I can start to increase its fuel production. Right now, the speed at which nitrogenase does these conversions is pretty slow, but we can learn a lot by understanding how this enzyme works. Once we fully understand it, we can start to improve how we use it. We might even be able to use nitrogenase to make other fuel products like gasoline. This system can serve as a way to recycle and reuse CO and CO2. And if we can use these little tiny biological machines to pull enough gases out of the atmosphere, we can reverse the devastating impacts of global climate change. Thank you.
All right, well, what got you interested in this?
So I've always asked why. I've always wanted to understand how things work. And for a lot of the important things that we need to do biologically, enzymes are the answer. And I had a great undergraduate professor at Willamette University, which is a little school in Oregon. And Dr. Fisher taught me everything about enzymes, and she got me really excited about what they can do and what we might be able to do with them. So that's how I kind of got into this general field.
Wow. And did you go right from Willamette to UC Irvine, or?
I took a year off. I kind of traveled around, spent some time on the East Coast, which was new for me. And then I spent 3 months working as a volunteer in Costa Rica, working for like a reforestation project. So I didn't do any science during my year off, but for some reason they let me in.
Tell us about what you like to do when you're not researching nitrogenase.
Well, I like to maintain an active social life, I guess, so I like to hang out with my friends. That's good, right?
That's good, yeah.
And I also sing, and I'm trying to learn how to play the guitar. So I like to go home and sit down with my guitar, sing, and play along.
What do you play? What do you sing?
Anything that gets caught in my head that has less than 5 chords.
C, G, D, A, E
I'm working
minor.
towards F right now. That's a tough one.
F is hard. You got to hold the 2 strings down. Right. So, when you sing, what do you sing?
All kinds of stuff. So I really like. Right now, I've been listening to the band Young the Giant a lot. No?
Anybody know them?
Also, Frank Ocean is one of my favorite artists. He's amazing, and I like to sing his songs because he's got a great voice. He sings in my range, so
it all works.
It all works, yeah.
Yeah. Where'd you grow up? Give us a little bit of your family history.
I'm from Oakland, California, originally, right? Yeah. So plenty of the people in this room work there. I love Oakland, and I grew up there with my mom, and I don't have any siblings, but I have a dog who might as well. Me and my sister. And then I went to
What's the dog's name?
Serena. She's a Rottweiler. I love her.
Okay.
And I went to a little liberal arts school in Oregon, Willamette University. Got really excited about chemistry and biochemistry there. Decided I wanted to get a PhD, and now I'm at UCI. And it's a great school.
Yeah. So you're having a good experience.
Yeah, I really like it.
Yeah. When you were in the 4th grade, what did you think you would be when you would grow up? Did you think, I'm going to study nitrogenase?
No, I didn't, and I didn't even really love chemistry until I got to college. But when I was little, I wanted to be a surgeon. But when I get nervous, my hands shake. So I thought that probably wasn't a good route.
Not a good fit for you. No, it's good to have that self-awareness, isn't it?
Right.
Yeah. So, and what about your future? Where do you see your future taking you?
I want to work for a biotechnology company, and I'd like to kind of serve as in a management position, maybe, kind of getting scientists together to solve big problems. And this is the one I care about the most. I want to save the world. I want to help solve global warming. And so I think that we can do that with science, but we've got a few more steps to take.
Right. Well, and particularly in the current environment, I might say. Yeah. So do you see yourself speaking out about science and about climate change as you move forward?
I'm kind of opinionated, and maybe you noticed, but I think that it's important to stand up for your beliefs, and I believe really strongly in science. So yes, I want to be an advocate. I want to, one of my role models is Neil deGrasse Tyson, because he's so good at getting on stage, getting people excited about science and making it approachable for people. And I want to be able to do that for the rest of my life.
Right, that's great. And is that what got you interested in Grad Slam?
I had a lab mate who had done this competition, and then my boss was kind of like, hey, you should try this. And so I entered, and it's been really fun.
Yeah. How was your campus competition?
It was good. It was wonderful. I'm always amazed to see people who are so passionate about all these different things. We're all devoting a little bit a few years of our life to these tiny little subsets of research. And people love it. They have to, right? Otherwise, we wouldn't survive as grad students.
Right.
And it's wonderful to see that passion come out.
What's the hardest thing about being a grad student?
You know, okay, at least I see this with science, is that science often involves a lot of failure. You know, so you go a long time without sometimes seeing success. So in some ways, this was a nice change for me because I won something, and it was a reminder, okay, I'm actually okay at things. But sometimes with science, you just kind of feel like you're not doing things right, but it's not your fault.
Yeah.
That's what I try to tell myself.
That's right. Well, you clearly did something right to get here, so congratulations. All right, so welcome to the stage our 9th contestant, Leslie Rith Najarian.
I'll sleep tomorrow. I'll exercise next week. I'll start dating after I graduate. These are some of the ways that students postpone their mental health. 1 in 10 students meet criteria for depression, 1 in 5 an anxiety disorder, 1 in 3 significantly impairing stress, and 100% of students experience these issues at some level because that's university life. So what we need is effective prevention programming that can teach students skills to improve their daily life. But, of course, there are reasons why prioritizing our mental health is difficult. Busy schedules, stigma, a preference for figuring it out myself. So, how is my research overcoming these barriers? I'm building an online anxiety and depression prevention program that students can use when they want, where they want. Now, there are some online programs that have been tested in research studies and they're effective, but they're not widely used because they're poorly marketed and they're especially not used by male students. On the other side, we're increasingly seeing consumer apps and products which are more widely used, but we don't even know if they work because they haven't been researched. My program combines the skills and engagement strategies of these research programs along with those consumer-oriented marketing strategies. So my program teaches skills week by week, interpersonal skills, physical exercise, mindfulness, and students log skills every time they practice. All of this is incentivized by things like weekly prize drawings, insights into their stress and mood ratings, and customizable email practice reminders. Now, I'm advertising this same program here under 2 different names, the Happiness Challenge and Reboot Camp. This is in order to investigate the impact of marketing on student enrollment. So, did Reboot Camp actually attract different students than the Happiness Challenge? Yes. The men. So, especially those from the physical sciences. So the exact same program is marketed, different names, different students sign up. Overall, more students were engaged. In terms of performance, my research studies show that the students who complete the program experience significant improvements in their anxiety and in their depression. 5,000 students have already tried this program, and I'm continuing to research how marketing and incentives can engage more and more students, because this knowledge could be applied to any mental health intervention. But shifting tone, at my college graduation, 2 seats were empty because 2 would-be 2012 grads had committed suicide before graduating. I like to imagine a world in which anxiety and depression are prevented before students get to that place. So this research isn't rocket science, but mental health is something that affects everyone on every day. And the more students we can engage in it, the better. Thank you. Thank you.
All right, so how did you get interested in this topic?
In mental health?
Yeah.
I mean, it really is something that affects everyone and every day. So I think that, I guess as a child, my brother was really mean to me. And I wanted to understand, why doesn't he like me? And why are people so different? And we're coming from the same parents, and yet we have such different personalities. And then, grow up and you realize that life's okay and it's okay that your brother is mean to you and you actually like him after all. But what did you do to get there? You
Right.
know, what were all the things you had to go through to kind of become who you are?
Right. And then how did you get interested in kind of the marketing aspect of this?
So I just have like this aversive experience, because I care so much about clinical psychology and mental health, that when there are these programs that are offered that should be good, but they're called something, like there's this intervention for teenagers called Cool Kids. And it's just something where I'm like, no teenager is gonna sign up for something called Cool Kids. And it just started like occurring to me that mental health has to be something that like is kind of cool or is funny or is fun, not just like serious.
So how did you pick the names for these programs that you were testing?
So originally, I was just running it under the Happiness Challenge, and it had mostly female participants, which is standard. Like females in general are more help-seeking. And so I very specifically was trying to think like, okay, if we're gonna come up with a kind of a partner brand, and we're trying to see if we can get more of the males. I was doing a lot of research on business. There's something called like fricative consonants, and so the more fricative consonants like t and b that there is in a name apparently is more masculine.
Who knows?
I know, and some focus groups, and so it was really exciting when it turned out that it did get more men. But then there were also other subgroups that I wasn't even looking for, like physical science students to slightly older students. There were a number of different things.
So do you have any hypotheses on what are the causal agents of this mental health issue?
For like the differences in the names?
Yeah.
Yeah, so there is some other research going to show that men typically are less direct when talking about their mental health, and so rather than saying I'm sad, they're like, oh, I'm not feeling so great. And so there's just something about like the language even, so like the Happiness Challenge is very explicitly like, I'm signing up for this because I'm interested in being happier. Whereas like Reboot Camp, it's a little bit like, oh yeah, I want to become more efficient or, build better habits.
Yeah.
Something like that.
Right. So tell us about your journey. How did you get to UCLA? And
Yeah.
what's your bio?
So I grew up in northern Minnesota, and I knew I wanted to go somewhere else for college, but I didn't know where. I ended up going to Harvard University for my undergrad, and that's really when I started getting interested because students would talk about stress there as if it were like a badge, like kind of just like, how much sleep did you get last night? Oh, I only got 3, I only got 4. And so I realized that like undergrad and grad school should be a time where you instead build positive mental health habits. And so I got increasingly interested in clinical psychology, and I worked at Boston Children's Hospital and then applied to grad schools and ended up coming to UCLA.
So you came to a warmer climate.
Yeah, it's been awesome.
Yeah. And what do you like to do when you're not doing research?
So I do a lot of rock climbing. My fiance, hi Eric, he is also a grad student and is also a rock climber. And so pretty much every Sunday, we try to go spend the day rock climbing and then go back to normal life.
Well, good luck.
Thank you.
All right, welcome to the stage our 10th contestant, Jeff Hollett from UC San Diego.
For women in the developing world, access to safe and effective birth control is an issue of life and death. Every year, hundreds of thousands of women die due to complications with their pregnancies, and the majority of those are happening in countries with the lowest birth control usage rates. That's because in a country like Uganda, there are significant cultural hurdles that women need to overcome before they can start using birth control. Many of these women aren't supposed to be using birth control, but a huge burden is placed upon them if they have to sneak around and hide their package of birth control pills every day for months at a time. As such, the most popular form of birth control in these countries is actually the injectable. This is something that women can administer themselves in private, and a single injection can give continuous coverage for up to 3 months at a time. Now, there's been a lot of research recently into how to better distribute those materials to women, but what's actually inside the needle hasn't changed for decades, which would be fine if the drug was perfect. But it's not. This drug took over 20 years to gain FDA approval in the United States, and when it finally did, it came with warning labels for severe side effects, things like irreversible bone mineral density loss. But those side effects could have all been predicted if you simply knew the geometry of the drug inside the syringe. These women are receiving injections of three-dimensional crystals of drug, which you can think of simply like a cube. But the problem with cubes is as they dissolve over time, they get smaller. Or more importantly, they lose surface area, which changes the amount of drug that's available to the woman's body at any given time. So that means the drug concentration profile in the blood looks something like this. At the beginning, the cubes are large, so there's a burst of drug, but it's followed by a long tapering tail as the cubes are getting smaller and smaller. But we still want the tail to stay above a certain contraceptive threshold level for as long as possible, so we increase that initial dose until the burst is so high, many women experience side effects. Now, my research is on how to change that geometry, to go from a three-dimensional cube to a one-dimensional wire. And I do so by using porous particles as my template. These particles have tunnel networks buried all throughout that can be filled back up with drug, so it's now protected on all sides except the top and bottom surfaces. These structures can only dissolve in one dimension, up and down, which I can concede sounds like a small change, but it has huge implications in terms of surface area. These structures can get thinner, sure, but they can't get smaller, which means that the surface area on the top and bottom is the same on day 1 as it is on day 100. So now the drug concentration profile stays flat and consistent with a sharp tail at the end, meaning women can still have continuous coverage, but none of the side effects. My hope is that this research will enable women to have access to safe and effective birth control, no matter where they live. Thank you.
Jeff, what got you interested in this topic?
Medical research in general, I attribute to my parents. Both of my parents are medical doctors, and all the time when I was growing up, I always assumed that I was going to be a medical doctor too, until I realized I was scared of needles. I can't even donate blood because my blood pressure spikes up. So doing medical research is at least as close as I can get to being a medical doctor without having to touch blood and things like that.
Right, right. Although this is injectable, it does require a needle.
Someone else can do that.
And tell us a little bit about your journey. Where did you grow up? Do you have brothers and sisters? You know.
Yeah, so I'm actually the only contestant that's not American. I grew up originally on the east coast of Canada in a town called St. John's, Newfoundland. I'm surprised I got any noise for that. People in Canada don't even know where it is. Yeah. But in general, it's a very blue-collar place, and there's very poor science literacy in that area. So I was always really interested in science and things like that, but I knew that ultimately I would probably have to leave. But now hopefully I can come back with skills to sort of beef up the area of science and medical research in Newfoundland eventually.
Right. So how did you make your way to California?
I did my bachelor's in nanotechnology engineering at the University of Waterloo back in Canada. And then all of my professors recommended that I move to the United States for graduate school. And it's funny, I'm in the material science department now at UCSD because when I was applying to universities, there aren't a whole lot of them that have nanoengineering programs. So my professors always told me, well, the equivalent is the material science program. And then I got to UCSD and realized it's one of the only universities in the US that has a nanoengineering program. I just wasn't looking for it.
And how have you enjoyed living in San Diego versus St. John's, Newfoundland?
It's funny because the first question has come up multiple times, is everyone loves the weather. No.
No?
I'm a big guy, I run hot. So it is, I don't like that sun. I get a sunburn if I'm out for lunch too long. You know, like I'm still inside enjoying that air conditioning life. But I mean, in general, the people here are great, and there's a really strong scientific community and collaboration and things like that. So as a school, I love UCSD. If you could move a little further north, I might appreciate it.
We could move the sun. What do you like to do when you're not doing research?
One of the few things that I do to retain sanity is I actually do improv. So I do improvised comedy. I get on stage roughly 3 or 4 times a month and just sort of do silly make-em-ups and stuff like that. I found that when I started grad school and I wasn't doing it, I was really annoying all of my coworkers and lab mates with dumb jokes and stupid bits and things like that. So having a creative outlet has helped not only my research, but also my lab mates.
So can you do a bit of improv right here for us now?
Nope. No, I cannot. Absolutely not. Nope.
No jokes?
Nope. No. No, I'm good with just the talk.
Yeah. Well, the talk was great. Thank
Great.
you.
Thank you so
Congratulations.
much.
Let's give a round to all the contestants. Great job, everybody.
Now the awards ceremony. And we're going to do this in the sequence that first the president will present the People's Choice Award, then we'll have the third place award winner, the second place award winner, and finally the grand prize winner who will receive the Slammy. And of course, all the presenters were extremely impressive, so we want to thank the judges too. They did a terrific job under very challenging circumstances. Before I invite the president up, we have apparently tried to add some glamour and glitz to this part of this ceremony because we want to demonstrate that Hollywood has nothing on the Bay Area. And so to help with our awards, we have a duo that oozes charm. We have Mr. Jeff Hollywood Hall over there. And well, we simply know her as Ms. Slammy. Is Ms. Slammy present? If you're interested in booking this duo, please see me afterwards. Maybe we can work something out. So let me invite President Napolitano back to the stage, please. And Ms. Slammy has the envelopes. Thank you. So this is the People's Choice Award. And it goes to Leah Foltz, UC Santa Barbara. Okay, now we come to the third place award. And this one goes to Jeff Hollett, UC San Diego. Second place award goes to John Feltz, UC Santa Cruz. And now, the moment we've been waiting for the grand prize award, the first place award. This goes to Leslie Rith-Najarian, UCLA. The Slammy. Now, there's one other aspect of this, which is that the dean from the campus with the Slammy winner is invited to come up, please. We have an award for Robin Garrett. And again, the president. So this is a plaque that moves with to the campus of the winning individual. It started at Irvine, is at Riverside last year. It'll move to Los Angeles. Congratulations. Okay. So thanks everyone. So it just remains to thank everybody on behalf of President Napolitano for coming here. We want to especially acknowledge LinkedIn for all of their fine support for this event. Alan Blue, Guy Berger, Katie Farrick, Joshua Kahn, and the entire audiovisual team, everybody here who really helped make this such a memorable event. We also want to thank our graduate program alumni who have very kindly come and provided advice and encouragement to our graduate students at UC. I want to thank the folks from the Office of the President, particularly Pamela Jennings and her team, for all of the help that they provided in pulling this together. So you can appreciate this was really, truly a team effort. And I think all of you appreciate just the energy and the inspiration that we all gathered today. And we look forward to next year's event. It'll also be exciting, and we hope you'll join us for that as well. So thank you very much.